Welcome to Science Unplugged, the podcast where science and conversation collide. Join us for a deeper look into the world of innovation. We are your host, myself Ankur.
And myself, Khushi.
Let's explore the insights from the world of science. From the lab to the field, we will dive you into the stories and experiences of researchers, experts, and innovators who are shaping our understanding of the world. Our guest today is Dr. Harbinder Singh. He is a postdoctoral researcher whose work spans synthetic organic chemistry, molecular modeling, enzyme assays, and medicinal chemistry. His research explores the development of novel molecules with potential against cancer, Alzheimer's, bacterial infections, and many more diseases. Welcome to Science Unplugged, Dr. Harbinder Singh.
Thank you very much,
Okay.
Ankur. Thank you. I'm sorry.
So
Go ahead.
can we start, Dr. Singh?
Yes.
Okay, so Dr. Singh, your journey from Punjab to GNDU, then California, then now to the University of Toledo was truly remarkable. So what was the aha moment that made you choose research as your life's path?
First of all, thank you very much, Khushi and Mr. Ankur. Actually, Dr. Ankur,
Thank you.
for inviting me on this very good platform. So, okay. So I'm a postdoctoral fellow at the University of Toledo here in the United States. So let's talk about my research interest. Okay, so I guess my interest basically started in the class of 11th or 12th when we were learning about the chemical reactions in organic synthesis and chemistry subject basically. At that time I was wondering like why, like how those scientists would know about the timing of those chemical reactions. Like how would they know like this chemical reaction will take uh, 3 hours, 4 hours, and some reactions taking too long, like days, like 2 days or 3 days. But how they would know, like we have to run this reaction for a long time, and it will complete after that time. Then this question was in my during my schooling days, but I could not get an answer during my school days. When I completed my schooling and I went to the College of Pharmacy in Amritsar, and joined a program in Bachelor of Pharmacy. And at that time, we had to, in a practical, in a class practical, we had to make a medicinal compound by using a single step chemical reaction. And at that time, I was little curious to know like how we will check the progress of the reaction. Then there is a technique TLC, thin layer chromatography, by which we can easily predict the timing of the reaction, like by which we can just tell like how much reactants are still there in that chemical reaction and how much product is forming there. Then according to that we can judge the timings of that reaction. Then I realized, oh wow, this is really a great, means the science is science. So then I realized with this theoretical knowledge we also need a practical as well as experimental knowledge. So in that way, like then the only way is to get a good knowledge is the practical as well as experimental things to do. Then the only way is the research to get the vast knowledge. So that's why like I went into the research, and after that, after B pharmacy, after Bachelor of Pharmacy, I went to Guru Nanak Dev University to pursue my uh, uh, masters, and after that I did my PhD. Yeah, this is how I started my research.
Okay, so it is good to know that if you really wanna understand the practical aspects of any theory, the
Yeah,
best way to go into the research.
yeah.
And uh, uh, I can see that you have actually spent nearly 5 years at Guru Nanak Dev University as a research scholar. So, what was the most transformative lesson or experience from the university that still guides your scientific approach today?
Okay, so basically, I spent 7 years there at Guru Nanak Dev University, not five, because I did my master's there for 2 years, and after that, I did my PhD for 5 years. So
Okay.
overall, 7 years I completed over there. Yeah. So okay, so the experience. Okay, I think the most important thing I learned from my PhD is, I can say, the importance of independency in research. So, like freedom in research. So, if you are free to do anything, then that is that is important in research. So, my PhD supervisor, Dr. Bedi, uh, he was that kind of person. So he gave everyone the freedom to do research. So he never denied my research ideas, and he also gave some suggestions to basically improve that ideas, right? But he just gave the freedom to explore the things in science. So in my belief, like the independency in research is the crucial thing,
And
especially
I
in
will
science.
just add on this independency also depends on your supervisor. So what you are telling our
Yeah.
audience that choose your supervisor wisely so that you can have a very independent way to conduct your research.
That's true. That's true. And with that experience, that independency, I learned a lot, and I also published research and review articles a lot, and even
Okay.
even joint corresponding author with my supervisor. So with that experience,
That's
I
great
came here.
actually.
I'm sorry,
I said that's great. Like if you're a, it's a great thing to have a joint
corresponding
co-author
author. Yeah,
with your own supervisor. It's a take.
yeah,
Yeah.
yeah.
Okay.
Then after that, like I joined the
Hmm.
University of Toledo, basically California first, and then University of Toledo. So here, with that experience, with that independency, today uh, I also give training to undergrad as well as graduate student. Yeah, so that experience really worked for me. Yeah.
Okay. Thank you very much.
Mhm.
But okay, so
And
my next question is that your research spans several complex areas in like organic synthesis, enzyme inhibition, molecular modeling, and structure elucidation. For our listeners, can you please explain how these pieces come together to form a bigger picture to the drug discovery?
really interesting question. Yeah.
Yeah.
So in drug discovery projects, uh, these areas combine to each other. Like all the areas have the crucial role in drug discovery projects. Let me explain one by one. So organic synthesis. So let's assume any of the particular disease condition. So first of all, we have to know the underlying mechanism of that particular disease, like which kind of proteins or which kind of enzymes are particularly involved or responsible for that particular disease. If we know those proteins or enzymes, then we can develop the inhibitors or like medicinal compounds. So inhibitors, medicinal compounds, drug are the same thing. Don't be confused in between these terms. Okay. If I say inhibitor, if I say drug, if I say chemical moieties, those are all the same things. So to inhibit those proteins or enzymes which are like overreacting in those types of conditions or medical conditions or disease conditions, then we have to inhibit those proteins or enzymes by making some of the compounds, some of the medicinal compounds. So how we can develop? By using organic synthesis, right? But what kind of molecules we have to design? Here is the role of molecular modeling or docking studies, we can say. So molecular modeling is basically the computational tools by which we can visualize the whole protein or uh, enzymes structure. So basically, protein or enzymes are basically the chain of amino acids, like tangled big chain of amino acids. That is protein or enzymes, right? So all the proteins or all the enzymes have different particular shape that is mainly responsible for that particular disease. So by those molecular docking tools, we can visualize those amino acids, those protein structure, those enzyme structure, and from there we can we can predict like which amino acids are mainly responsible for their function. So from those proteins, few amino acids are means functionalized. So that amino acids make a binding pocket or active site binding pocket. So by those molecular docking tools, we can develop those molecules that can perfectly fit into those binding cavities or binding site or functionalized binding site. Then we can predict, oh, this kind of molecule we need to perfectly fit into the binding site, and it can inhibit the function of those proteins or enzymes. So this is the main function of those molecular docking tools. So yeah, so organic synthesis, molecular docking, the enzyme structure, enzyme means proteins or enzyme structure analysis. These are all interlinked to each other for the drug discovery projects. Yeah.
It seems very simple that suppose if I wanted to cure a disease, what you are telling our audience is you first have to know which enzyme or protein is responsible or
Yes.
associated with that disease, and then you have to find a drug molecule to prevent the mechanism of action
Yes.
related to that disease. And you use something known as computational and molecular modeling to
Mhm.
know which drug molecule is going to bind to the
Those
enzyme.
proteins.
So
Yes.
how good is this prediction? Like, am I gonna get a prediction which is 100% accurate, and then the life is easy, or is there something which
That's
is not
very
that
good question.
straightforward?
So intellectual thinking. That's very good. So okay. So thing is, the molecular docking is the tool just to predict the binding pattern of the molecule with the enzymes or proteins. Sometimes it does not work. Sometimes it's perfectly work, right? So the molecular docking tools in these days are like at the advanced stage these days with the fact of artificial intelligence. So artificial intelligence basically what it do it from the previous database from those protein inhibitors or enzyme inhibitors, it collect the data from there, and it will perfectly reshape all those molecules, and it will just tell you means these compounds can be perfectly fit. I'm not saying like this will be 100%, it will give you the exact compound that will definitely work, but all those are computational tools, right? So sometimes it happens, sometimes it works, sometimes not. Yeah.
So it is just an helping tool for us to get
Yes.
some potential
Some basic ideas like,
candidates. And
yeah.
then you are saying that ultimately a scientist has to do many experiments to figure out whether those candidates are working well or not.
Yeah, after all those things, like after developing the molecules, then we have to test those molecules for that particular proteins or enzymes by using in vitro methods first, and after that, if in vitro methods pass those molecules, and after that we have to do in vivo research like animal studies, and after that we can tell that these molecules have really the potential to inhibit those proteins or to work for that particular disease. Yeah.
Okay.